Hi everyone and welcome back to my channel. I'm Aman Praveen and today we'll be discussing about the NK cells reacting in the innate immune responses. So as you know, innate immunity has NK cells in it. You see, lymphocytes, there are three categories. First one is the B cells, the T cells and the NK cells. These B and T cells act in the acquired immunity, whereas the NK cells act in the innate immunity. So this is one peripheral smear, and you can see the examples. The images of this is the neutrophils. This is the small lymphocyte. This small lymphocyte can either be a B cell or a T cell, and you see all these granulomatous structures, very condensed. This is the basophils, and so let's understand the morphology first of all. You see these B and T lymphocytes are there, right? These are basically small lymphocytes. They look small and have a lot of nucleus in it, whereas the NK cells are larger cells and have granular structures present in these. You see all these dotted structures in it, and the cell is comparatively larger than the B and T lymphocytes. So these NK cells, just like any other cell, has receptors, and these receptors may be CD16 or CD56. This CD16 molecule contains the Fc receptor of the IgG. As you know, in an immunoglobulin, it has heavy chains and light chains, right? And in these heavy chains and light chains, it can be this can be divided into variable compound fragment and a constant fragment. Okay, this constant fragment can otherwise be called as an Fc component, and this variable fragment can be called as FAB component. Okay, so that is what we are saying here. The CD16 molecule is a receptor for the constant fragment. You get it? The constant fragment. Okay, so yeah, and the CD56 is an NK cell adhesion molecule, which helps a lot of cells to adhere together and fight off a much larger sized antigen. What happens is actually, you see, these NK cells can't actually directly go and bind to the antigen actually. So what they need is that these antigen have to be combined with an immunoglobulin. Since they have an immunoglobulin receptor, they need this antigen to be structured together with an immunoglobulin or an antibody. So this antigen antibody complex occurs, and this thereby goes and binds with the Fc receptor of the NK cells. This is the Fc receptor, and see, it goes and binds with the antigen antibody complex. This then what happens is that this NK cells destroys the antigen. So this can go in two ways actually. See, what happens is that a particular surface structure, for example our own cells, have their own complexes. Like in a normal cell, it also contains antigen, right? Which can be carried out by the MHC1 class receptor. So this is a normal cell, and this shouldn't be actually destroyed. So what happens when there's an MHC class 1 receptor? This antigen goes and binds to the MHC1 class receptor. Okay, this is the antigen and this is the MHC1 class receptor, and this when it this NK cell comes near it. An NK cell has one activating receptor and an inhibitory receptor, and when it's a class 1 MHC, the inhibitory receptor goes and binds. So as soon as the inhibitory receptor goes and binds with the MHC, what happens is that this NK cell does not get activated. Therefore, this NK cell does not go and kill the cell. Whereas in a virus infected cell, what happens is, these viruses actually inhibit or depromote the class 1 MHC. Therefore, the virus infected cells are class 1 MHC negative cells. Therefore, there is absence of an MHC1 molecule. So what happens is, the ligand present in that cell goes and binds with the activating receptor. So what happens when the NK cell gets activated? There is expression of the NK cells, and thereby it kills the infected cell. So this is how NK cells interact and kill virus affected cells. So this is a brief about the innate immune responses and about the NK cells. I hope you enjoyed this video, and I hope this was informative. And thanks for watching, and make sure to click the subscribe button, make sure to click the like button, and if there's any doubt at all, just let me know in the comments. And as always, be awesome.